Thank you, Dr. Asnaki. And thank you to the organizing committee and also to our Australian hosts. It's a great honor to have this opportunity to present results from our analysis of stem rust collections in Kenya. I'm speaking as a representative of our collaborative team on this group project. I am one of 10 co-authors, all of whom made important contributions to our group effort. It's a great pleasure to work with Pablo Oliveira, Ruth Wanyera, Sam Gale, Doug Luster, Sridhar Bhavani, Dave Hodson, Matt Rouse, Les Sabo, and Eugene. The outline for this presentation is in 4 sections. The first 2 sections provides background information on stem rust sample analysis and races identified between 2008 and 2012, and a description of the stem rust outbreaks that occurred in Kenya during 2013 and 14 on varieties that had previously been resistant in the field. The next 2 sections present our results of phenotypic and genotypic characterization of isolates from 2013 and 14, and evaluation of seedling resistance to the Kenya races. Here's an overview of our stem rust sample analysis process. 2 USDA biosecurity labs coordinate to receive samples 12 months during the year. The primary lab is a cereal disease lab in Minnesota, and this is where the race identification and other analyses occur. The Foreign Disease Weed Science Research Unit in Maryland receives samples in the off-season. During March to November, samples are received and increased at FDWS in Maryland. This capacity to increase samples immediately after receipt has greatly increased the chances of viable samples. After increasing, the samples are sent to the cereal disease lab, where they are processed in the months between December and February. That's the winter in Minnesota. Single pustule isolates are derived and race typed based on the North American differential set. And the next steps include confirmation of the race, genotypic analysis, and additional phenotypic characterization. Races that were identified during 2008 to 2000 collections from the Najaro Nursery and Farmers Field were based on 111 isolates that were derived at the CDL. And all of these isolates from these 5 years of collection were identified as races in the UG99 group. 33 isolates were identified as TTKSK. This is UG99. 76 isolates were identified as TTKST, and 2 isolates only found in 2008 and only at the Najaro Nursery were identified as TTTSK. This table presents the North American differential set, and I'm presenting it to highlight the genes that are important for the rest of this talk. Race TTKSK is virulent on 17 of the 20 North American differentials. Race TTKSK is avirulent only on SR36, SrTmp, and SR24. For this project, we included an additional source of TMP, which is shown at the bottom of the table, Triumph 64, which is the donor of SrTmp. Returning now to the races identified between 2008 and 2012, the summary table shows the 3 races along with the first year of detection in Kenya and differences in virulence. As I described before, TTKSK is avirulent on SR36, TMP, and 24. TTKST, which was first detected in Kenya in 2006, is virulent on SR24, and TTTSK, first detected in 2007, and in our samples only again in 2008, is virulent on SR36. The stem rust outbreaks that occurred in Kenya in 2013 and 2014 occurred on varieties that had previously been resistant in the field. And prior to that, in 2011, 2 new varieties were approved for release in Kenya, Eagle 10 and the cultivar Robin. Robin is postulated to carry SR2 and SrTmp. Both Eagle and Robin had been shown to be resistant to all known races in the UG99 race group. And farmers who planted Robin reported very high yields and large harvests before 2013. Robin was and still is a very popular variety in Kenya, and that's because it's high yielding. In 2014, nearly 50% of the wheat area was planted to Robin. Stem rust caused severe damage to Robin. There were reports of 60 to 70% yield losses in unsprayed fields. And it's the smallholder farmers who cannot afford to apply fungicides who feel the most impact from these stem rust outbreaks. In response to the stem rust outbreaks on Robin, stem rust collections were made from the wheat production region. The top map shows the general wheat production area in Kenya, and the bottom map shows the 2014 survey sites where stem rust was detected. Samples collected in these years were sent to the USDA ARS labs and also to the Global Rust Reference Center in Denmark and the Agriculture and Agri-Food Canada Center. And cross-validation among these different rust labs is very important and very valuable. Now, with that background information, the rest of the presentation will talk about phenotypic and genotypic characterization of the isolates from Kenya and evaluation of seedling resistance. 4 viable samples were collected from 2013 collections, resulted in the isolation of 2 different races. In the summary table, it shows that the 4 field samples all were identified to have race TTKST. The top field sample was a mixture of 2 different races, TTKST and TTHST. The race TTHST had not previously been reported, although in 2014 a very similar race, TTHSK, was isolated at AAFC by Tom Fetch. Race TTHST is similar to TTKST, except for that it produces a low infection type on SR30. You can see the low infection type on SR30 in the photo, and the high infection types on SR24 and 31. And I'm including SR31 because of the importance of this virulence. Now the summary table includes the new race, TTHST, and its unique avirulence on SR30. It is also virulent on SR24. 24 viable samples from the 2014 collections resulted in isolation of 4 races. They're shown in the summary table, along with the number of locations where each race was found and the number of single pustule isolates for each race. The 2 new races identified in 2014 are shown at the bottom of the table, TTKTK and TTKTT. It's important that these races were found at multiple locations. These races, as you have heard, were also isolated at GRRC from collections in Kenya and other African countries. And again, this cross-validation among labs is very important. Race TTKTT is similar to TTKST, except high infection type on SrTmp and TRIME64. The photos show high infection types on SR24, 31, and TMP. And in a separate inoculation at a separate time, we have photos of high infection types on SRT and TRIME64 next to it. The summary table now has the 2 2014 detected races at the bottom and their unique virulence on SrTmp. One of the races is avirulent on SR24, and one is virulent on 24. In addition, part of our phenotypic characterization includes inoculation of the new races on varieties and cultivars grown in East Africa. And here I'm showing the results for two varieties, Robin and Digalu. The first lines show the seedling infection type after inoculation with the new race TTKTT, and it shows high infection types. In comparison to the seedling infection types produced by TTKSK and TTKST, which are low infection types. Robin is widely planted in Kenya still, and Digalu is widely planted in Ethiopia. Both of these are postulated to carry SrTmp. This table shows the seedling infection types produced by 5 races on 17 lines. It's a lot of information. I'll summarize it by column. The 17 lines are primarily monogenic for important stem rust resistance genes. They're part of a set that we call the universal resistance set. These genes are resistant and remain effective against the UG99 races. The two columns, the first show the seedling infection types, are the reactions produced by TTKSK and TTKST. These low infection types are very similar to the infection types produced by the 3 new races. So these genes, these 17 genes, remain effective against these new races. So there are sources of resistance available. One last point about this table is it shows a lot of phenotypic similarity between the new races and known UG99 races. But this phenotypic similarity by itself is not enough to show genetic relatedness. For that, we need genotype data. So shifting to the genotypic characterization, it's analysis that was conducted by Les Szabo and Jerry Johnson. And there's additional information in the poster by Szabo and others. 20 isolates from the 2014 collections and 15 additional reference isolates were genotyped using a custom PGT SNP 3K chip. After filtering the data, 1,634 SNP loci were polymorphic and used for the analysis. A phylogenetic tree was constructed using neighbor joining method. This tree shows 35 isolates, 15 reference isolates noted by an asterisk, and 20 isolates from the Kenya collections. So first I'll go through the reference isolates and explain the clades. 3 of the reference isolates are representative of the UG99 race group, which is Clade 1. Clade 2 includes isolates from Yemen in the race JRCQC, Clade 4B, and then jumping down to Clade 4A at the bottom, are isolates from Ethiopia that are race TKTTF, or the Digalu race. This race has virulence to SrTmp, but it's very distinct from the UG99 race group. And Clade 3 is represented by Yemen reference isolates as well as Ethiopia isolates that are race TRTTF and RRTTF. These races also have virulence to SrTmp. Now the 20 isolates from the 2013 and 2014 collections clustered very tightly with the 3 UG99 isolates in Clade 1. So this is definitive evidence that the new races are genetically related to the UG99 races. And focusing just on Clade 1, the UG99 race group, the isolates are color-coded by race. So the 3 new variants are shown by the colors of the 3 bottom colors on the key. And the important part of this focus on Clade 1 is that there is subclustering within the Clade 1, and it is not associated with race phenotype. So there are genetic differences within a race as well as between races. The final set of results I'll show use the new isolate as a screening tool for seedling resistance evaluations. Selections of lines from 4 different international nurseries were screened for seedling resistance to races TTKSK, TTKST, and the new race TTKTT. The 4 nurseries include the Kenya Advanced Material, US Winter and Spring Wheat nurseries, and the CIMMYT stem rust resistance screening nursery. In total, 1,055 lines were evaluated. The results are shown here by nursery. The first column is the total number of lines screened for each nursery, and next are the number of entries that had combined resistance to TTKSK and TTKST. And then the numbers are shown out of those with the combined resistance, the number that are susceptible to TTKTT, the new race. So for example, the Kenya Advanced Material had 80 entries with combined resistance to TTKSK and TTKST, and of these, only 18, or 23%, so not only, I should say, 18 or 23% were susceptible to TTKTT. In the winter wheat, the US nursery, 44% that had been resistant to TTKSK and TTKST are susceptible to TTKTT. And that's a total of 91% in the US spring wheat nursery. For the CIMMYT 10th stem rust resistance screening nursery, there were 119 entries with combined resistance to TTKSK and KST, but only 2 were susceptible to TTKTT. And another important point is that Eagle 10, which was released at the same time as Robin, shows seedling resistance to TTKTT and also showed resistance in field conditions. Considering these results and results that we've heard from others here today, implications for the future include that stem rust sampling efforts have provided critical data to inform the development and deployment of host resistances. And the deployment of single major resistance genes is not an effective strategy in East Africa. Varieties with additional sources of resistance other than SrTmp are needed. We'd like to gratefully acknowledge many individuals and our funding sources. Godwin Macharia was actually in Minnesota and helped with some of the race identification and is now at Calro. And we also thank the staff and drivers at Calro who make the surveys possible. We gratefully acknowledge Melissa Carter, Gordon Cesar, Jerry Johnson, Sam Stockson, Janine Lukes, and Nick Prenevost. And without the contributions from the USDA ARS, Durable Rust Resistance in Wheat, administered by Cornell, and the University of Minnesota, this work wouldn't be possible. Thank you.